next up we've got Alexander Bucksch from the University of Arizona since not too long ago, and we are moving from above ground to the hidden half below ground. Looking forward to your talk.
Thank you, Bettina. So first of all, thank you for letting me talk here. It's an awesome meeting so far. After, I think, every 10 years, it's in the Americas and the first time in the US. So it was just pretty cool to have that here. It's not the full hidden half I want to talk about. Where do I have to put that here? Yep. So, it's not working. Now, it's not the full hidden half I want to talk about. It's actually about the root hairs, right? The ones that are missing here on the screen. But it's great to have roots now. So that is a huge improvement to other backgrounds I had in my life. But so these are these root hairs that you see here already on the title slide, and they're just very important. And there's, I think, over the last 2-3 years, or this project is already going on about 5 to 6 years. There's actually always a paper like this. This is just 2 weeks old. Yeah, root hairs, they're important. You never look into it, but they have a lot of role in combating abiotic stresses and drought and these kind of things. And there's a lot of potential we cannot really unpack and capitalize on. So I give that up to others. You can read these or other papers. I want to just jump into very basic things, like can we actually really count them? Right, if you want to map them or whatever, you can have a very basic question. Can we actually count all of these root hairs? So the top one is a bit difficult. The lower one, probably your 5-year-old at home can do. That's rice, pretty easy, very short, not so dense root hairs normally, most often. And the top one is corn. That's I think it's a Thai variety from microlaboratory Kwan in Thailand. And it's getting really dense. So it's hard to even guess how many root hairs are there, right? And then if you keep in mind, they're very thin. Even under a microscope, it's hard to see what this single elongated cell actually is and to discern it. Okay, so we thought it would be nice if we have a method to just start counting. And we started off like this. So we have these microscopy pictures of stained roots. You have a lot of possibilities when you have a microscope, right? You can search for a certain plane to have always accurately the most vertical. You're basically always having the same plane you're imaging so that the images become comparable. And what we did is we just use elastic to train a standard random forest model to kind of classify root hairs from the main root where it's coming from in the image and from the background. And sometimes we put a noise class in it if the microscopy was not too correct. But that's how we get that. And then the question is, how do we get from such a resolved image to individual root hairs? Now that there this problem starts. So what we do, and I just put here now a comic for a second, we can use the medial axis, which is a skeleton, to just get lines through every root hair, right? But they are connected, they have intersections, and we're starting off having this problem. Which intersections belong? How do we cross these intersections to get to a full root hair? You see that here on the bottom, there are many, many solutions, and then the many solutions that we have to choose from. So we have to look at all these possibilities to select the real root hair that you want to measure at the end. Okay, so there's a black box, it's called simulated annealing that can do that. And that's what we did. So this should be a video. Yep. So basically it relies on minimizing 3 terms that we put together. It's the curvature, it's the number of root hairs that we currently have, and then there is a term which is how far is that segment that we had away from the main root. Simple terms that we minimize by just randomly checking which segments between intersections belong together or not. And slowly, step by step, you actually resolve all the root hairs that are in your image. And you see on the side, these are the functions how they just minimize. So now it just sprinkles around a little bit at the end. Okay, pretty easy, right? the student is graduating soon. The paper is, if you want to see the paper on that method, it's just in JXBot, as a Journal of Experimental Botany. It's in the accepted papers. You can download it already. It will come out in December, right? Okay, what do we really get? So you see in a defined area that is in focus, we can count all these root hairs in the same plane. And we have now a lot of possibility, right? We can say, okay, it's that long. Now we have a scale to it, we can measure the length, and we can also give you density measures, which are common measures that breeders are interested, that are physiologists interested, and many other people. We thought, okay, let's do that. Let's get millimeters for the density, or hairs per millimeter, and let's get the length out. So the first thing we did, so one of the students of our collaborators actually started taking a bean picture, a rice picture, or 5 bean pictures, 5 rice pictures, and 5 corn pictures of these roots, and started counting every single one. Right? It takes a while. And what you see on the top is simply how the distributions between manual count and automatic count by the software differ. And then this is just the number up there, and then the root length on the top. But then if you look the correlations between the 15 images and the averages, there is some correlation, but also some bias in it. Especially if you go to all the right in the middle column, which is out there, it's the density where there are huge discrepancies between what the manual person measured in the density and what the computer measured. Even so, we have actually every hair in hand. Last row is just a large data set where we had thousands of images of bean. We got that from Mark Libaud here at UNL back then. He's now at Mizzou. And we just looked, hey, how far are we off? And you see pretty much there is a huge bias. The more root hairs you get, the stronger the bias gets. Let's unpack that a little bit more. On the left side, this is simply the first 3 are common bean. The first 3 bars, the second 3 bars are rice, and the last 3 bars are corn. Now, it's just a sanity check. Rice, pretty short hairs in our data set. Common bean was in the middle, and corn has this huge variation and very long hairs. That's kind of what we see in these data sets that we had. But the interesting part is that we see, depending on which species we look at, we actually see different biases arising between the measurements, even so they equally nice detect every single root hair within the focal plane that we analyze. You see there is, depending on the measurement, there's always quite a bias. And interestingly is also the spread, and keep that in mind for later. The spread actually in the common bean is a bit higher. There are some outliers that we initially couldn't explain. But there's generally for the length, we get very good correlations. Now, let's look one further to the density. We saw already that might make some trouble. That's happening. That makes a lot of trouble. And from our perspective, we see that there's a clear bias in it, but it's even more than a bias. It's even not a correlation sometimes, because you cannot guess how many roots are in there, especially for corn. It looks very bad, but also already common bean is fully off. And that has to do with how these roots curve around, these root hairs curve around, and you saw all these intersections that suddenly root hairs go from right to the left because you put them on the imaging plane, on your microscope. So you run into these things when you just look at it, and you will miss it. So your density measurement will be off because you cannot decide, is this root coming from here or from there? It's just getting to that. So there's a lot of subjectivity that can now be repeated over and over again if you just have an algorithm that does the same thing. Okay, let's see why that works. I think this is the bigger question. And this works, this algorithm, because we always assumed, yep, we have a length, and this root hair is tubular, right? Like a tube, just very long. And you see here also in the back, like how they, on the microscope, just curve around, really shoot all the way through your image. So that's something you can just not grasp by eyes. But yeah, the underlying assumption is that they are just long tubes. That's what we had also in the algorithm. And we can measure the length along that tube as a central line. Gives you also another question. There's so many outliers, right? So are they really all tubes? If you think about this is just one cell that extends from the epidermis. Above ground, we have a similar situation with the trichomes. Sometimes they're multicellular, but most of them are actually unicellular. And they show this huge variation. They're not all tubular. And for root hairs, we have a few, in the beans, you have the crook, so before it nodulates, and a few mutants that are out there. And extreme stresses can induce kind of a split in the morphology of your root hair. But there's by far not as much known as about the trichomes. So what we did is we looked a little bit deeper, especially into the bean, and we found the following, and that as a hypothesis, we already brought it out, that we should look into that in this paper down here, in Current Opinion. And that's a work of my student Ankita and Ellen, that is somewhere here in the room, and our collaborator, Mark Libaud, that is now at Mizzou. We saw already very early on in development on the radical, actually, something emerging below ground that never comes up during development. It stays below ground all the time. And it looks like a trichome on the right, kind of, but very different than a root hair. But it stays there. And we wondered, what is that? Some people told us that at the beginning, a few years ago, must be a trichome, you're doing something wrong, right? That's what you do. What happened is, we just put it first into hydroponics. And said like, okay, let's stress that out with a modified Hoagland solution to induce some stresses. And we see suddenly that this hook thingy that we have there, or call it hooked hair, which is also just one cell, as you see, has a stress response very similar to a root hair. Basically the same, right? It becomes denser, it becomes longer, depending on the stress. So seeing that under the microscope was for us enough. Okay, we measured already the length. We have that as a central line. We can do that with the same shape. But we want to look a little bit deeper. So we want to also look, is this the same shape like a trichome, or is this the same shape like a root hair? Obviously not, but there must be a difference in it. We should be able to quantify that. So we did the same thing again. Background, a noise area around, and the hooked hair here that we have, and we used elastic just with a random forest model to classify that, and we look at these red parts. Okay. One thing you do is it's a Procrustean analysis that we do. In fact, if somebody heard about it, it's just basic shape statistics, but what happens is you have an average shape that we just determined based on the traits we could measure with our tool before. We just took the average hooked hair that we saw in this early development, and then we overlaid them, aligned them as good as possible in their centroid, took the outline, and then we used something that is called iterative closest point algorithm. If you're computer vision folks in here, that's a fairly standard method to align or find the best possible overlay between two shapes. So you do that by you norm the outline to the same length, and then you get the last result. Now, and from here, normally you would look at landmarks that we don't have on that shape. So the only difference we do here in the comparison of the shape is to say, use something with what's the Hausdorff distance, which is every pixel on the outline that we have. We measure the distance to the closest pixel on the other outline. Sum that up, average, done. That's our distance, that's our measure of similarity between the two. If they match perfectly, it will be 0. If they don't match so much, then you will have big numbers. Works pretty linear, is a standard method. So that's what's coming out. So what you see here is very, very much up there. What we did is we used these radicals at the beginning. They were about 3, 4, 5 days old. We just sorted them by length and put them into 3 classes in short, middle, and long, just to account for differences in development. And it's turned out that the higher the older the plant gets, the more similar the shape becomes. So it becomes no difference in the overall form of these root hairs anymore. But still your length is changing. That should now tell somebody something. They cannot be. right? It cannot be if you have either a trichome with diffusive growth or a tip-driven growth of a root hair. Cannot be that result because both of them would either change the shape or the length, right? But that's not happening. The shape stays constant. So that was enough for us to think, what should we do now? So look at these single cells, and luckily we have also single cell sequencing now. So we decided to go to this, back then it was a lab in Nebraska, where we said, let's grow these plants and then shave off these single cells. There's a method to just shave off them. And we used the trichomes on the epicortal, fairly above ground. We took root hairs, and we took this new hooked hair from the plant. So these are different locations on the plant. And what we did, what you just saw also from Jakub, we did a Go analysis, right? Just the gene ontology, and then it came out. So BG means hooked hair, what I called it before. That's just the labeling we had in that experiment. It was below ground. And we saw there's a lot of things going on that have to do with nutrient uptake, with resource uptake, with water uptake. In fact, later on, we found out there's also quite a bit of the suberin biosynthesis involved in this hooked hair, in this BG cluster, or in this set that we have here. Well, the others do fairly what you find in literature. Very different functions are associated with it. Let's think a little bit more. Okay, we have all this single-cell sequencing data, right? Started just doing the next step, which is compare that to orthologs in Arabidopsis, in common bean, in orthologs in Medicago and Arabidopsis. And you see that the BG, this hooked hair, if you compare it to the trichomes, has a very different expression compared to the orthologs, compared to the trichomes that we had above ground. And the same happens with the root hair. So if you look at the difference in expression, you see it's almost opposite to the trichomes, even though it has the same shape, the same form. So that is even more reason to look even deeper. And we did this. So this is just the U-map of the common bean root. It's also constructed from a lot of orthologs. I think most of you are familiar with that. And each point in this picture is one cell. So what you see is that the root hairs are associated with this cluster number 5. And does this have a? Not sure. Yeah, it has no pointer. But up here, this peninsula on the top, that green thingy coming out, are 2 clusters that are associated with root hairs, definitely. And that's known. And there are some that are just more generally classified as, oh, that's some epidermis, something. So what we did is then quick sanity test, right? Look at the expression we have from these hairs that we shaved off. Turns out the root hairs, yep, they map directly in this cluster number 5 and 6 up there, these red ones. That's where you expect them. That's what literature tells you. But if you looked at this hooked hair, and at the trichomes, yeah, they should not show up so much. They are above ground. But this hooked hair showed a very strong preferential expression in a cluster number 4. And 4 is all the way out there, if you see that, under these dark blackish, dark green appendices. So this doesn't work, this thing, but it's kind of here. Okay? So it's a very isolated cluster that you actually see there, and we just mapped it then as a very last step. We mapped everything back onto the U-map, so that's what you see here. That's the root hair. Now the sanity check for the trichome, yeah, it's everywhere a little bit, but it's basically just noise. And you see that out there is that hooked hair in the cluster that has not been described before, with a little bit of noise similar to the root hair. So we know there are definitely different functions, especially resource uptake associated with it. And that brings out for us the big hypothesis that there's probably an epidermal cell type we never looked at. I think that this is very likely. That's how far we are right now. For us, that was a big deal. And I just want to say already thank you. And hope that you get something out of this talk, which is there might be more out there than we think, as things we can maybe use to push crops a little bit further, things that we didn't know even existed. Also, the other thing I want you to take away from this talk is we can actually measure these complex things like root hairs, even if you are not able to do it by eye. I think it's a very important thing to get in your mind, to say there's things there that we don't know about it, and let's search for that. Okay, if you're more interested, because I'm running out of time, into the aspects of the suberin biosynthesis or more details on that topic, so Alan, my postdoc, has also a poster there with a few other aspects of that study. Just find them, it's a cool poster. I say thank you to all the sponsors that in some way contributed to that, and the whole lab that is here. Thank you.
Thank you very much for an exciting talk, and I'm curious to know what you will name those hooked hairs, whether they'll get a special name to mark them in history. Do we have any questions? At the back.
Thanks for a great talk.
Oh, Valerio, hi.
Hey. So in bees, we know, generally speaking, that there is a big shift between the Mesoamerican pool and the Andean pool.
Yeah.
I know that that's too, at the population level for your study here, but I just wondering if you had any insight as to how early root development might be influenced by this big divide that we are aware of.
All right, I didn't tell you the genotype here. It's actually, I think, one from Michigan. It's L8857 that is coming from your future university, so you have that available. And welcome to the US then. We saw it also in other common beans, all across the common beans we have available in my lab. There's some are from the Andean, some from the Mesoamerican panel. So we saw it in both. I cannot tell you if these 30 varieties we have overall, it might be just a selection that is a lucky shot. I don't think so, but I can tell you we didn't see it in soybean. So we tested that already. But there's something you can transfer or up to breeders, right? That's why I have you.
Do your root hair hooks show gravitropism? Do they always hook down?
No.
One more question over there.
Oh, thanks for the nice talk. The images were all in front of a nicely clear background. Do you have any indication how it looks like when you are in real soil?
Again, what background?
A nicely clear background, so you didn't see any structures in the background where you have imaged the root hairs.
Yeah.
But when it's in real soil, it might be different because of the different cultivation systems.
Okay, I didn't tell you the environment, that's true. So there was one part was a hydroponic study, the first one, but the whole transcriptomics we did in real soil grown plants. So that was here at Nebraska in the facility.
Oh.
Okay, if there are no more questions, then thank you very much, Alex, for a really exciting talk.
Thank you.